Today I am going to explain about melon fruit fly. Melon fruit fly are scientifically known as Bactrocera cucurbitae. These are pest flies and they mainly infest plants which belong to family Cucurbitaceae. Under this family, hundreds of vegetable plants come, and this pest fly is known to infest a number of them. More than 100 different types of economically important plants are infested by this pest fly. Some of them are listed here, like pumpkin, bitter gourd, bottle gourd, watermelon, long melon, cucumber, sponge gourd, musk melon, snake gourd, squash melon. These are about 10 economically important vegetables which are grown in our area, in our country, and they are all infested by this insect pest. About 80 different plant species are reported from our country, that is India, which are infested by this pest species and cause profuse economic losses. And more than 100 species of plants are listed which are infested by Bactrocera cucurbitae or melon fruit flies. If you see the morphology of the insect fly, you will find that the two sexes, males and females, are sexes. It means it is a dioecious species, and there is distinct sexual dimorphism. The male individual possesses a rounded abdominal area or tip, whereas the female one possesses a protruding part in the posterior abdominal portion, and that is called as ovipositor. Now this portion ovipositor, which is being shown in this diagram, can be more elongated and more pointed in the posterior side when the female is ready to oviposit the eggs. So you can see the two flies. The female is a little bit larger than the male individual, and if you see the gross structure of their wings, you will not find any major difference between the wings of the two sexes, whereas the abdominal tip can only be used to differentiate the two sexes, and it's quite easy. Even with the help of naked eye, the males and females can be easily identified. Now this species belongs to phylum Arthropoda. Its class is Insecta, order Diptera. Diptera order that includes mainly flies, that is housefly, mosquito, Drosophila, Chironomus, and a number of pest species. They belong to Diptera because it contains large number of insects. And there is a family Tephritidae under which this genus Bactrocera comes, and there are more than 40 different species of insects are there which come under family Tephritidae. Bactrocera is one of them, and a species which is melon fruit fly is cucurbitae. So Bactrocera cucurbitae is melon fruit fly, and this fly is quite big menace to the farmers, to the agriculture persons, because they face much loss. So you can see the taxonomic position of this insect fly, and now we have actually observed the pecten hairs in case of male. See, besides the abdominal tip portion, which is mainly used to distinguish the two sexes, we tried to see whether other structures are also there which can be used for sexual differentiation, and we found that in case of male, in the mid abdominal portion, see, this is a dark area, and on this area, on this side, you can see the presence of hairs, and these hairs are actually divided into two halves, that is right and left halves, and such hairs are referred as pecten hairs. They are found only in case of male. A female individual completely lacks this structure. So it is written here that pecten hairs are found on the dorsal surface of abdomen of only male individual. The pecten hairs present on right and left half of abdominal portion can be counted to test whether this trait in males show continuous pattern of distribution and fluctuating asymmetry. At least these two phenomena of evolutionary biology could be tested by using the number of pecten hairs present in the individuals of this species. See, we have been working on this fly because of its economic importance. We selected this fly to work, and we thought that we must develop certain strategies so that we can control this fly and the economic loss could be prevented to large extent. So laboratory culture stocks are here, and we use actually large plastic jars to culture these flies. We introduce males and females in these jars and keep some moist cotton from which these flies can take their water content. We put in small petri dishes sugar which is mixed with yeast powder, so that forms the food of the adult flies. Besides that, we also put small pieces of pumpkin, and females which are ready to lay eggs, they actually pierce their ovipositor into those pieces of pumpkin and lay their eggs. So by this way we have been able to increase the number, because we collected only few flies and also larvae from the infested fruits, and then brought them into the laboratory, put them in the jars, and a time came when we were able to have large number of flies in these jars. So we have now plenty of flies, and we can work on different aspects of this pest species. You can also see the life cycle because working on this species is quite interesting, and before starting its major studies, one should be aware about its different life stages. These are eggs. The eggs are actually white in color and they measure nearly 2 millimeter in size. They are almost flat or a little bit concave in the ventral side and convex in the dorsal side, and there is presence of micropyle also that indicates the anterior portion of the egg. Then in about 24 hours from these eggs, larvae come out, and these larvae then undergo molting and change into second and third instar larval stages. They measure from 7 millimeter to 11.5 millimeter in size. Those larvae which are almost ready to pupate, they will be maximum in size. They are third instar larvae and they will be measuring around 11.5 millimeter in size. And you can see the black spot, that is the anterior side of this insect. It is the chitinous structure that is used for feeding purpose. So this is actually the biting and chewing kind of mouth part in this larval stage, and it takes nearly 8 to 10 days or even sometimes more to change this larva into the pupal stage. But see, the third star larvae, when they are ready to pupate, they will be jumping out from the fruits and they will be coming into the swale. So swale is must for their pupation purpose, and then they penetrate into the swale, which will be the area where they will be pupating will be around 0.5 centimeter to sometimes 10 centimeter or even 15 centimeter deep into the swale, depending on the texture, on the type of swale, because that swale which is very compact one, in that swale the larva will not be able to penetrate in more deeper portions. But if it is sandy one, then for protection purpose it will go in more deeper and moist areas so that it can pupate over there. And the most surprising thing is that if swale is not available for the larval for the larva to pupate, then it will dry up. It will not be able to undergo in the next developing stage. And in about 8 to 10 days, the adult fly will come out or hatch out from this pupa. So it takes around 12 to 28 days for completion of its life cycle, and this also depends on the life cycle stages, developmental stages, and the completion of its life cycle. That completely depends on the environmental condition. Definitely when the temperature is low, at that time the developmental period will be longer, and when temperature is high, then it will take lesser time to change into the next stage. Then here you can see the eggs have been collected in the petri dish, and these are white in color, elongated structures. So these are a large number of eggs. A single female, can lay nearly 1,000 eggs, but all these eggs will not be laid in a single fruit. Rather, a female will be visiting fruits, and even the soft portion of stem are used for depositing eggs. And in a single area, in a single bunch of 5 to 10 eggs will be laid, and the female will go on selecting appropriate areas on the fruits stems, and rarely in the roots also, where she can deposit eggs. But a single gravid mature female can lay thousands of eggs during her lifetime. See here, it is a small piece of pumpkin in which the larvae are growing. You can see this is a third instar larva which is present over here, and this is another larva. This is third larva, the fourth one is here, fifth one is buried half in the fruit, and you can see here these are the portions where tunnel-like structures are there. It means the larvae have gone inside, they have moved here and there, and because of their movement, such burrow-like structures are visible. So if they have infested the fruit, then that fruit will not be consumable, it will not be palatable at all, and they make actually such fruits unusable. So this is the drawback, because sometimes the fruit gets highly infested with the larvae, and such fruits are actually thrown out. See here, these are pupae which have been collected from the swell, and these pupae are put on a paper. Those which are about to undergo pupation will be light in color. See, in this case this one is light in color, means a larva has just undergone pupation. But then those which have attained advanced stage, in their case you can see the eyes are showing blackish spots over here, but these are dull red structures of the pupae. It means they are quite advanced stage of pupae, and as I told you that it takes 8 to 10 or 12 days for a pupa to complete its life, and then it will hatch out into adult flies. This is a world map showing the areas where very high infestation of this melon fruit fly is reported. Entire United States, that is the portion where any adjacent islands are also the places where the infestation of this pest is there. Although this Bactrocera cucurbitae, its native place is Indian subcontinent, and from this place it went to different areas of the world, and the entire Asian countries, even total Australia, some parts of Arabian countries and Africa are actually affected by its infestation. Although it is worldwide in distribution, that is, it has been reported from every part of the world, but these are the places, means colored areas are the places from where very high level of infestation is actually reported. Then the insect, this particular fly is of significance because it is of economic importance, and we must be aware about its life stages and its behavior, so that we can control its infestation. And it can be cultured in laboratory conditions, and some people culture it for larval purpose. These larvae are used to feed fishes and other animals. so that way it can be used in other for other purposes also. Being a dipteran insect, its polytene chromosome preparations can be done, and all sorts of chromosomal aberrations or things related to polytene chromosome observations can be performed. Then it possesses a very short life period, and it produces a large number of progeny, so it can be used for studying genetical aspects. The mature melon fly males are attracted to several chemical attractants because a mature female releases pheromones. So chemicals similar to such pheromones are known and could be used to attract males like anisyl acetone, raspberry ketone, and zinzerone. These are the chemicals which are actually used to attract the male individuals, and if they are attracted, then they could be used for study purpose, or they could be killed so that the females will not be able to mate with them. So this is another important use of it, that by using some chemicals they could be trapped. They are good pollinators as they visit to different flowers to feed. So this is the importance of these flies, that besides causing much damage, they are considered to be only useful since they visit a number of plants or flowers and help in pollination process. We are actually working on the life history traits of this fly. Purpose of working on this aspect is to know the different aspects of its life history, and by knowing details of such things, we can design, we can plan certain strategies so that we can control this fly. So what does this life history and life history traits mean? Life history denotes the life of an organism from its birth to its death, describing the age and stage-specific patterns of maturation, reproduction, survival, and death. That comes under life history aspects. So there are certain events that are directly connected to the life of an organism, often called life history traits, which depends on the physical and ecological environment of the organism. There are 7 traditionally recognized life history traits. These traits are size at birth, means when the individual actually hatches out, or even the different stages of its development like egg, larva, pupa. These aspects could be considered under life history traits. So size at birth, then growth pattern, age and size at maturity, number, size and sex ratio of offspring, then age and size specific reproductive investment, age and size specific mortality schedules, and length of life, that is the life span of the individuals. All these 7 features come under life history traits, and we have selected some of these features to know about its such traits and also behavioral aspects, so that the knowledge of those things could be used to eradicate and also control this insect fly. Thank you.